Hi, my name is Christopher, and I'm here with my team partner, Tuan. We did this project together, analyzing the binary interval search algorithm. And I've linked the paper to the top left corner, and I've also linked to the Git repository. If you're interested in that, I'll be sharing the slides also. So before we get into the algorithm proper, I wanted to give a background on why the algorithm is important and why other genomic algorithms are important and what they're used for in the first place. So imagine we take a blood from a human being. This blood is taken to the lab and processed. And after the processing, they're able to sequence this blood, which is to turn it into repetition of letters, the AGTC. And this letter holds information about every organism, how tall they'll be, their eye color, and things like that. And in humans, we have over 3 billion base pairs of this information. Now, over the years, the way to sequence data has sort of like increased, and storing this data in different file types have also increased. Now, how do we sort of like extract information from this? Extracting information from 3 billion base pairs of letters is difficult. So what happens is this chromosomes intervals are extracted from this 3 billion base pairs. So these chromosomes could be of any length, maybe any chromosome of interest. Say, for example, we wanted to know why someone is of a certain height, and we sort of like want to extract just that particular chromosome and look at it. And that is sort of like the point of it. So in most files, the nucleotide are saved in the chromosome and then the start location and the end location. The start location helps us know where, and the end location, both of them helps us know where this chromosome fits into the large scale of things. So when we sort of like want to compare it with other chromosomes, or when, so that we know in what particular location did we get this strand of nucleotide. So yes, in most files, the chromosome ID just includes the interval that we're interested in, the start location, and the end location. So this is an example that I've put up. So assume that we have a patient 0 that has a cancerous gene, and we want to determine if this gene is rare or common. So if the gene is common, that means that we will see it in a lot of the database. Say, for example, we're checking the population of everybody in Boulder. If the gene is common, we'll see that gene repeated in most people. And if the gene is rare, we'll also see that the gene will not be repeated in most people. So a way to do it is by finding matches. So assume this reference genome, the one on top, is our chromosome of interest, and assume the sequence read is the chromosome of everybody in the population of all the of people in the database. And then when we look through the reference and the database genome, we're able to find matches. Sometimes it matches and sometimes it doesn't match. Now we see other algorithms also that do string matching, but the difference between this is we're not just matching the string, we're also matching the location that it is, because strings could actually match, but they could not be in the same location. So having the strings and having the location and is very important for genomic algorithms. So I've talked a bit about interval intersection, but what does the basic question that interval intersection asks is how much of our reference genome actually matches with the database? So when we have the patient 0 and we match it in the database, how much of the database actually match of our reference genome? And having algorithm to do this is very important because it's a way to discover new genes, is a way to discover new diseases. It's used in precision medicine and so on. And there are lots of algorithms and systems that do this. We see the UCSC genome browser, we see Giggle, which are all algorithm and platforms for matching nucleotides. So we get to the binary interval search algorithm. And the binary interval search algorithm helps for finding and counting the number of intersections between two sets of genomic intervals. So when we have our set A, which is our query set, and we have our set B, which is our database set, it helps to find the match and then able to count the match. And then the binary interval search algorithm uses the start and end coordinates the way the genomic data works. But this is used in a very different way, which we're also going to explain as we go forward. And then we're now able to count the number of the intersections. And this executes in O of n log n, where n is the number of intervals. So we'll also analyze the complexity further as we move on. So the binary interval search algorithm asks very specific questions, and these questions sort of like helps understand what the algorithm is doing. And the first one is decision. So when we look at two sets A and B, the first thing we need to do is to decide if there is a match to begin with. And when we decide if there is a match, then we can move on to the second one, which is to count the total number of intersections between the sets A and B. And we have other sort of like minor functions that also works behind sort of like the interval counting and enumeration. But for the purpose of this project and for sort of like the importance of the algorithm, we'll be focusing on just the decision and the counting because that's where the bulk of the algorithm is on. So I'll be handing over to my team partner, Tuan, to start explaining the algorithm.
Thanks, Christopher. So the core argument of binary interval search is that instead of using the inclusion definition to search for a number of intersecting interval, they use the exclusion definition. So to be specific, in naive algorithm, intersecting interval set I contains all of the interval pairs A and B if A star before Bn and An before B star. This is gonna take O n square to check for all the pairs. However, in the binary interval search, given that we have a database B and a interval query AI, the set L gonna contain all of the interval in database B that ended before AI starts, meaning that the interval in B that is to the left of AI. Similarly, for the set R, gonna contain all of the interval in database B that started after AI ends, meaning all of the interval in B that is to the right of AI. And then interval set I is simply B exclude L and R. Because we can quickly count a number of interval in set L and R, we can count a number of intersecting interval in I a lot faster, resulting in n log n time complexity. So the BIS algorithm is based on one fundamental function, I count, which determines the number of interval in the set B that intersect query interval AI. So data set B is split into two integer lists, BS and BE, which are sorted in ascending order. Next, two binary searches are performed because BE is sorted list of each interval and coordinate in database B. The element with indices less than or equal to the last in BE corresponds to the set of interval in database B that end before AI starts, aka completely to the left of AI. Similarly, the elements with indices greater than or equal to the first in BS correspond to the set of intervals in database B that start after AI ends, aka to the completely right-hand side of AI. From these two values, we can directly infer the size of intersections in set I. So using ICANN algorithm, we can perform each query independently, which leads to the parallelism property later on. So the intersection decision algorithm leverage the I count earlier. So first, we need to sort BS and BE. Then for every interval in the query set A, we check for a number of intersecting interval If the result C is greater than 0, the answer is yes. Otherwise, the answer is no. Similarly, for the interval intersection counter algorithm, we accumulate the intersecting interval across the query set A and then return the total number of intersecting interval C afterward. So for the I-count to work, so this is the time complexity analysis. For I-count to work, the interval set B is split into two sorted integer lists. This is gonna take O B log B for a sorting to happen. So each binary search take O log B in the I count. Third, I count gonna take O log B to perform. Now, for each query AI in the query set A and the reference database B, the search gonna take O A log B. So generalization of the dominant term gonna become O n log n. Now I would like to hand it over to Christopher.
Okay. So to conclude, we've talked about the genomic sequence, the idea of the genomic sequence, which is looking at nucleotides, looking at specific parts of the nucleotide, which can be known as, it could be chromosomes, it could be a gene, it could be a lot of things, but sort of like just looking at very specific intervals. And we can find information about these intervals by looking at other intervals and finding the similarities between the sets of intervals. And BEATS is an algorithm that is able to find and count these intervals. And BEATS uses the exclusion technique, like Tuan has explained, which is when we have an interval, we find everything leading up to the start of that query and everything after the end of that query. And by taking those things away, we're left with the things that actually match the query. And this makes it able to, the idea of parallelization, that we're able can have as many queries as possible and run everything at the same time. And this sort of like leads to the time complexity with the total time of the length of A or the length of B log B. And what this means is at each run, the length of A, depending on how many AI we have, could be bigger than the length of B, which is the database. Because for all the elements in A, or for all elements in all query elements, say for example, A and X, A is not equal to X. So we have for A is not equal to A, X is not equal to B, is not equal to C. So for as many query as we have, they're all different, and the length of the query could actually be bigger than the length of the database. So that's why we have this length of A or the length of B log B, which when reduced would now be n log n in total. And concluding on the I-count analysis, which is sort of like the core function of the algorithm. So we saw different iterations of the algorithm to do different, to solve different problems. But for all iterations of the algorithms, what remains constant is the I-count. And the I-count is, it helps to find the intervals directly without actually counting the intervals by using the exclusion method. And just by using the coordinates, we're now able to infer what the match is and now able to count the problem. So thank you so much for listening. Tuan, do you have any last words?
Yeah, so thank you so much for the effort you guys put in throughout the semester. We believe that we have learned a lot about analysis algorithm, and I think it's gonna help us a lot in our PhD journey later on.
Thank you so much. See ya.